Good afternoon to all. Today we are going to study a new virus, that is the rabies virus that causes the disease rabies. Actually, in this lecture we are going to take this lecture into two parts. The first part we are going to study the details of the virus, the morphology, its antigenic characters, the types of rabies viruses, and pathogenesis and clinical manifestation. In the next lecture, we are going to study the laboratory diagnosis of rabies, prophylaxis, and treatment. So, starting with the topic, first the classification and history of rabies virus. We will be studying the introduction. This virus actually is a bullet-shaped virus, which is an enveloped single-stranded RNA virus. Now basically, according to the morphology, it is a rod-shaped virus where rhabdos, which means rod. Now this virus actually belongs to the Rhabdoviridae family, which is classified into two genera, vesiculoviruses and the Lissa viruses. In vesiculoviruses, actually we are not going to go into the details of vesicular stomatitis virus, but definitely Lissa virus, which consists of rabies and rabies related viruses, we are going to see the details. Now, rabies virus, morphologically, as we have discussed previously, it is a bullet-shaped enveloped virus with unsegmented, linear, negative sense, single-stranded RNA virus. Now here is a diagrammatic representation to understand the morphology of the virus, and this is the electron microscopy picture of the virus. According to the morphology, it measures of about 180 by 75 nanometer in size. Shape is bullet shape. One end of this is conical, here you can see it's conical, and the second end is planar or concave. Okay. Now the outer layer, which is a lipoprotein layer, consists of lipid and protein. The outermost layer is the lipid layer, which is derived from the host cell membrane, where the virus is multiplied inside the host cell. Whereas the inner proteinaceous layer, it is actually coded by the virus. The viral genome has the genes to code for this protein. It contains glycoprotein spikes on the surface. Here in the diagrammatic representation, the green colored spikes, they represent the glycoprotein, which are present on the surface of the virus. Now these glycoproteins are said to be the spike proteins. They are also responsible for attachment of the virus to the host cell surface. Okay? Then the matrix protein lies beneath the envelope, and this fills completely the inner viral structure. So, this matrix protein actually towards the planar surface, it may get invaginated to form a concave shaped structure, or sometimes it may protrude out to form a knob-like structure, which is called as the bleb. Okay? Then the nucleocapsid and the nuclear material. This is a single-stranded RNA virus where the RNA is coated with the capsid protein. And so the nucleocapsid, again, it is a RNA genome and a RNA polymerase, which is called as RNA-dependent RNA transcriptase. So this becomes the total structure of the virus, where important structural components are the glycoprotein spikes, the inner membrane or matrix protein or the membrane protein, and the nucleocapsid protein and the RNA. Resistance of the virus. This virus actually is very sensitive. It is sensitive to ethanol, iron preparations, quaternary ammonium compounds, soap, detergent, and lipid solvents such as ether, chloroform, and acetone. Now these viruses, they are inactivated by phenol, formalin, beta-propiolactone, ultraviolet light, and the sunlight. It dies at room temperature and it survives for weeks at 4 degrees Celsius. And this virus can be preserved in a crystalline form at minus 70 degrees Celsius or by lyophilization. Now the antigenic properties of this virus. As we have discussed previously in the morphology, the outermost surface glycoprotein spikes, they are very important in the pathogenesis of the virus as well as the virulence and immunity against rabies. It mediates the binding of virus to acetylcholine receptor in the neural tissue. This is the most important fact which is responsible for the first step of pathogenesis, where the virus has to adsorb to a particular cell receptor. Here the cell receptor is acetylcholine receptor on the neural tissues. So the first step of pathogenesis starts with the glycoprotein spike. Okay? Antibodies to this, they are protective and stimulate cytotoxic T cell mediated immunity. This is a serotype specific antigen, the glycoprotein, and its purified form may provide safe and effective subunit vaccine. This is very, very important because this is the only vaccine which is said to be most safe against rabies. Because this is a subunit vaccine where the glycoprotein spike has to be extracted from the virus, it should be purified and it should be stabilized, and then it can be given as a safe vaccine against rabies. The rest of the vaccines, they do have certain side effects or certain contraindications because they are either prepared in neural tissue or in some other host where certain components of the host, they may play a role in the interfere in the immunity of the host. So this becomes the most effective vaccine against rabies. Then other antigens present in this virus, the nucleocapsid protein induces complement fixing antibodies which are not protective. So these do not have any role in the protection against the viral infection. Third is it is a group specific antigen, cross-react with some other rabies related viruses as well. Other antigens include the two membrane protein antigens, the glycolipids and the RNA dependent RNA polymerase. Now these antigens or antibodies to these can be used for the detection of infection, but not for confirmation. Rabies, the disease. Now the natural host of this virus actually is wild animals. Many wild animals, they are the natural host of this virus, and human infection is acquired from the bite of those wild animals, particularly fox and jackals, which bite the domestic dogs. And dogs which are bitten by these rabid animals, they bite human beings. So this is the mode of transmission how it has arrived from the wild animals to human beings. So the domestic dog which have been bitten by rabid animals, rabid wild animals, they transfer the virus to human being by bite. So this actually is a zoonotic disease which is acquired from animal host into the human being. So the natural host, they are cats, vampire bat, skunks, cattle, fox, jackals, and all these are highly susceptible to this infection. Whereas fowl, pigs, horse, donkey, monkey, fish, arthropods, they are less susceptible to this infection. But definitely, when a rabid animal bites these animals, they do get infected with rabies. Dogs and human beings, they occupy the intermediate zone between highly susceptible, highly sensitive and less sensitive animals. Pups are more susceptible than adult dogs. Mice is said to be the most susceptible animal to this infection, and they are very often commonly used for experimental inoculation for isolation of rabies virus. Now, basically, when we say rabies virus, there are two types of rabies viruses. One that is isolated from natural infection from a host, either wild animals or human beings. So that natural virus which is isolated, which is most pathogenic, it is said to be the street rabies virus. Okay, now this, when it is inoculated by any root, can produce the clinical disease rabies, leading to fatal encephalitis, with a long incubation period of 1 to 12 weeks. Whereas, in this case, with respect to street rabies virus, after infection, if you collect the brain biopsy, we are able to detect the presence of intracytoplasmic inclusion bodies. Inclusion bodies are the bodies present inside the cytoplasm of the infected cell where virus multiplication is taking place. Now such inclusion bodies which are present in the cytoplasm of the brain cells of infected host, they are called as Negri bodies. It was discovered firstly by Adelchi Negri, so it is called as Negri bodies. Now these street rabies viruses, they are not used for the preparation of vaccine because they are the wild strain of rabies virus. On the other hand, the second type of virus is called as the fixed rabies virus, which is an altered form of street rabies virus. Now during this alteration, after several intracerebral passages in rabbits, the street rabies virus undergoes certain changes and turns into a fixed rabies virus. Now this virus actually is more neurotropic but less infective through other routes. That means when it is inoculated into the CNS, it is more neurotropic, but when it is inoculated from some other side, it is less effective. Intracerebral inoculation produces fatal encephalitis with this virus also, with a short incubation period of 6 to 7 days. Then, Negri bodies, they are not seen in animals dying with fixed rabies virus. This is very important for you to understand because fixed rabies virus and street rabies virus, they are different from one another. One which is isolated from the wild animals as a wild strain, that is street rabies virus, and fixed rabies virus which is adapted to human being more neurotropic when it is inoculated into the CNS. This fixed rabies virus can be used for vaccine preparation. Now, the host range and the growth characters. How this virus infects, what are the different requirements of the host or the virus. Chick embryo, here we can inoculate chick embryo for the growth of this virus, and the site inoculated is yolk sac to obtain the yield of the virus. Second, serial passages lead to attenuated vaccine strain like Flurry and Keller strain. Now in yolk sac, when the virus, that is the fixed rabies virus, when it is continuously passaged into 40 to 80 eggs or 150 eggs, the virus is attenuated. Its pathogenic character is decreased, but its antigenic character is retained. Such a strain is called as the Flurry and Keller strain. Now this Flurry and Keller strain, which is propagated in the yolk sac of chick embryo, can be used for vaccine preparation. Now this strain adapted to duck egg, which is called as high yield of virus is obtained, used for preparation of inactivated vaccine. Now such vaccines, they are used only for animals, for vaccination of animals. Next is tissue culture, where we can grow the virus in the tissue cultures as well. Chick embryo fibroblast cell culture or hamster kidney cell culture can be used for the inoculation and obtaining the growth for vaccine preparation. Cytopathic effect may not be apparent, and the fixed rabies virus, it is adapted to grow in human diploid cell strains. When it means strains, it is a diploid cell strain. It is not haploid. Next is primary chick embryo cell culture and purified Bureau cell cultures. These are the different cell cultures which can be used for propagation of fixed rabies virus for the preparation of vaccine. Now the disease rabies. Rabies definitely is an infection which is transmitted from rabid animal, particularly in human population. The wild dogs or the street dogs, they are usually infected with this virus infection during fights with wild animals. And such a rabid animal, when it bites a human being, the virus is transmitted through the saliva of the rabid animal to the human being. So, dog bite is the most important method of transmission, where the animal is mad. Rabidus is a Latin word which means mad, and rabhas is a Sanskrit word which means frenzy or violent anger. Now, in this case, everybody knows that in rabies infection, the most important clinical manifestation is the fear for water, which is described as hydrophobia. Now, this is a very common feature which is seen in rabies, the human host which is infected with this virus. But what actually is responsible for creating this particular clinical manifestation of hydrophobia is to be studied in the pathogenesis and clinical manifestation. Okay? Now, hydrophobia, actually it means rabies disease in human, which is called as hydrophobia, is because of the patient exhibits fear for water and being incapable of drinking those subjected to intolerable thirst. The patient feels thirsty but is unable to drink liquid. That is the important feature of hydrophobia. Now, this peculiar feature is not seen in animals. It is seen only in human beings. Transmission. Human infection is by the bite of rabid dogs or other animals. The saliva containing the virus is deposited in the womb. Okay? And rarely non-bite exposures can also transmit this infection, particularly from domestic animals which are infected with the virus but may not show any signs of rabidness. Such transmission is by leaks of such domestic dogs, which may transmit the infection from the dog to the human host. Or even during the working in surgery, during transplantation, if a person suffering from an infection, if the particular cornea is transplanted from the infected person to another, the infection may be transmitted through non-bite way. Now, the virus is infected tissue may be transmitted to other hosts. Now, this is actually the way how it is transmitted. The rabid animal usually bites at the particular sites of human being, and mostly the extremities are exposed to the dog bites, particularly the hands. You can see the bite at the particular site, and the foot. Very rarely it may bite even the face, neck and back. So these are the common sites where the rabid animal attacks a human being. Now, during this attack, the bite is so deep that the teeth of the rabid animal buries into the muscular part of the human being, of the host. And during that bite, the virus which is present in the saliva of a rabid animal, the saliva is deposited deep into the muscles of the site at the bite. Now, this virus which is deposited into the wound, it penetrates the nerve endings and through the exoplasm reaches the spinal cord and the brain. Here is the diagrammatic representation how the virus enters the CNS. This is the muscle fibers where the virus has been deposited, which is shown in blue color. It enters the peripheral nerves and through the axon, it is carried to the neuronal bodies. Now this is how it travels from the site of bite to the CNS up to the dorsal nerves. Now here when it reaches the CNS, it rapidly ascends towards the brain to infect the brain. Now the virus ascends rapidly to the brain, multiplies rapidly, and there is centrifugal spread along the nerve trunk to different parts of the body, including salivary glands shed in the saliva. Now this is the complete pathogenesis of the virus, where the virus is deposited into the muscle fibers. These viruses enter the peripheral nerve endings, enter the dorsal root ganglion where they multiply. Now this is the site where they are traveling through into the CNS, and when it reaches the CNS, it rapidly ascends towards the brain. Now in the brain, this virus multiplies in the hippocampus and the cerebellum, which is the most important site for multiplication of this virus. Now this traveling from the muscular organ, from the site of bite to the brain, is called as centripetal spread from the periphery to the brain, where brain is said to be the central organ of target organ. Now after multiplication in the brain, the virus later descends downward to the different parts of the body, which is called as centrifugal spread. And this centrifugal spread starts through the nerve trunks from the brain. Now initially it enters the nerve trunks and reaches the cornea through the nerves, then to the salivary glands. And in this way it travels through the nerve trunk to the different parts of the body. Okay, so that means the virus is, during centrifugal spread, the virus is spreading from the brain to the facial nerves first, to the cornea, salivary gland, and then descends downward to the different parts of the body. Okay, now this centrifugal spread may be interrupted with the death of the patient at any stage, because the brain is completely involved and each function of the brain is completely lost, because of which the typical features of madness may be seen in the host. That is the important characteristic feature which we are supposed to record in the patient, where the brain loses its complete functionality. Okay? Now centrifugal spread may be interrupted at any stage by death. Now the clinical features. In human being, the incubation period ranges from 1 to 3 months, though it may be as short as 7 days or as long as 3 years. Now this incubation period actually depends upon the site of bite of the mad dog to the brain. The distance the virus has to travel to reach the brain. So that varies according to the distance between the bite and the brain, which is the distance virus has to travel to reach the brain. So it may be shorter when the patient is bitten near to the face, and it may be longer if it is on the distal parts of the body. Generally, incubation period is shorter in children than in adults. The course of disease, rabies, in human may be classified into 4 stages. First is called as the prodromal stage or prodromal phase, then acute encephalitis phase, coma, and death. Now during the first prodromal phase, symptoms are seen as they are seen in usual viral infections. In most of the viral infections, these are the common features seen initially in a patient, which represents viral infection, such as fever, headache, malaise, fatigue, and anorexia. Now, after this phase, the early symptoms which develop are due to the involvement of the virus of the peripheral nerves, where the bite has taken place. During this period, in 48 to 72 hours, what actually is seen is neuritic pain, which indicates the involvement of the peripheral nerves. Paresthesia and fasciculitis. Slightly there is a tingling sensation of the nerves, which indicates that the virus is trying to penetrate the nerve endings. So these are the features which are seen initially in 48 to 72 hours. Till then, the virus has not yet entered the nerve endings. Okay. Now, during this period, at the same time as the nervous system is being involved, some features of neurological complications may be seen, particularly apprehension, anxiety, agitation, irritability, nervousness, insomnia, and depression. Apprehension is where the patient feels fear about something. He may have the stimulus or he may not be having any stimulus, but still he has a fear. Secondly, anxiety. He is slightly anxious about whatever is going on. He may get the stimulus, but he may not respond to that. Agitation, irritability. The patient becomes irritated for anything, even if there is a stimulus or if there is no stimulus in front of him. So irritability is another important thing. Nervousness. He feels nervous about everything, about his disease, about the surrounding, about anything. The patient feels nervous. Insomnia. He is unable to sleep at night. He remains always in a, that is, he is not able to sleep during night, and he is depressed. Depression is another important feature which is seen in rabies. These last for 2 to 4 days. At the same time, some other features may be seen particularly in males. Excessive libido, periapism, and spontaneous ejaculation may be seen in some of the patients. Then, now at this stage, the person is being involving the CNS. And when the CNS is involved, it reaches the second stage called as the acute neurological phase, where there is hyperactivity in the patient. The higher functions, they slowly start decreasing their normal function or altering their normal function, during which the patient becomes hyperactive. A stimulus may be present or may not be present, but the patient becomes very hyperactive, shows bizarre behavior, bouts of bizarre behavior may be seen, agitation may be seen. Agitation may be seen, or seizures appearing between apparently normal periods. The pathognomonic feature in this case is difficulty in drinking liquids, even if the patient has intense thirst. Now remember, this is a very, very important thing which indicates that the brain has stopped its normal functioning. Okay, he may swallow dry solids but not liquids. Next, during the acute neurological phase, what happens? How water cannot go down the alimentary canal. This is the important feature which is to be understood. Attempt to drink brings such a painful spasm of the pharynx and the larynx, producing choking and gagging, that the patient develops a dread for even sight or sound of water. So the pharynx and the larynx, there is a choking because of which the liquid does not flow downward, and immediately it is thrown out from the mouth. And that is the important feature which is called as hydrophobia. Okay? Now, apart from hydrophobia, most of the cases they even show photophobia and aerophobia. Photophobia, where there is fear for light. The patient does not want to see the light. And even aerophobia, slight aeration may even irritate the patient, which is called as aerophobia. Now, these are the common features which are seen during the acute phase of encephalitis, which indicates intense inflammation of the encephalus and the brain, which alter the normal function of the brain, giving rise to madness. And ultimately, when the brain is completely losing its capacity, the normal function, the patient starts developing generalized convulsions. And this convulsive patient may further lead into the third stage called as coma. Survivors of acute neurological phase, they lapse into coma, and death usually occurs after 1 to 6 days due to respiratory arrest during convulsions or other complications. Now, as there is choking and gagging of the alimentary canal, the larynx and the pharynx, during the same period when the patient is respiring, both of these, the respiratory system and the digestive system, they are not coordinating properly with one another, and that's why there is respiratory arrest and the convulsive pattern may start developing. And during this convulsion, it may lead to death of the patient. Now, there is another important feature which is often seen in the persons who are handling the cases of rabies, particularly the nearest relatives who are taking care of the patient, or the persons in the hospital who are taking care of the patient. If they have a fear about rabies, many times they may develop a psychological disorder which is called as Hydrophobia or Hydrophobiophobia, where there is a psychological disorder in some persons exposed to real or imaginary risk of rabies. When they are taking care of the patient, they may think that the saliva of the patient might be having the virus which has been transmitted to yourself, and you are being infected with this type of infection of hydrophobia. So this is actually an imaginary consideration of the person who is taking care of the patient, and he may develop an imaginary risk of hydrophobia, and such a person may show the typical features of hydrophobia even if he is not being infected, and that is called as Hydrophobiophobia, or it is also called as Hydrophobia. Now, in this case, the patient may also develop features such as he may be afebrile, but he shows anxiety, irritability, and exaggerated hydrophobia. Now, such a person who is not been infected but still have the features of hydrophobia may be taken care of by sedation. And reassurance are generally the helpful to get rid of this feature. Reassure the person that you are not being bitten by any organism, any virus, or the patient. You are completely normal, you don't have any features of rabies. In this way, we can take care of such a patient. So that's all for what we are here today. The clinical manifestations, rabies virus, the structure and morphological details, and the antigenic characters. The next lecture will be on laboratory diagnosis and prophylaxis and treatment of rabies. Thank you.